everyone, for inviting me and having me here. I'm looking forward to this. Been looking forward to it since Robin invited me. So I'm going to tell you a long tale about assembling a continental fauna in Australia, mostly of vertebrates. And I'll focus mostly on amphibians and reptiles, just because those are projects that we've been working on recently that I thought would be fun to talk about. I imagine it happens something like this with, frogs on a little boat ending up in Australia. It's a bit more friendly if we think of it that way. I usually try and include a slide that tells you about how I ended up in Australia. I'm originally from New York, and usually it's, it takes 30 hours to fly there or whatever, but I found this thing online that will tell you the distance through the Earth between any 2 points on the planet. It actually, like, if you go to the website, it spins around. So it's actually, it's something about around 12,000 Ks between New York City and Canberra, if you were able to dig through the center of the Earth. And probably the reason I ended up in Australia, the best reason, is that I really think that the vertebrate animal fauna in Australia is really amazing. Growing up in New York City, there's not like that much going on. And so I tried to simplify this for you by doing a Google search, but you can tell the Google algorithm is completely cooked, because if you search animals of New York City, in the top hits you get snowy owl and fin whale and painted bunting, when the top three should certainly be rat, cockroach, pigeon. I don't know what else. But you can see some of these things. So how did I end up here? Well, I wanted to kind of work on this big question of how a continental fauna assembles. But Australia is a really amazing place for this, because it's a natural laboratory for evolution. The continent has been drifting around in the ocean, heading up towards Asia, and after it's separated from Antarctica for 20, 30 million years. So it's had a long time for radiations to evolve in situ, and there's lots of replicates for this. So we can start to ask questions about the replicated process of evolution. And in laboratories, sometimes things go horribly wrong. Like this turtle frog, which might be the world's worst frog. They can't hop. All the things that frogs do. They can't hop. They barely crawl. No tadpoles. Something is terribly amiss here. But sometimes things in the lab go terribly right, like with dasyurids like antechinus's, where this Ed Young title for this article is just too good to pass up. Animal has so much sex it disintegrates. I love that. So all of these things, I think, are what make Australia such a fantastic place to study vertebrate evolution. So how does a continent become populated? I'm going to give you 3 steps in this talk about how we get there, steps to succeed in Australia. And I just want to focus that I'm talking about animals here, and this is obviously a bit satirical. So 3 steps to succeed. The first one is immigrate. The second one is compete. And the third one is be flexible. And I'm going to give you a kind of use case from each one of these. And I'm going to start with frogs. Immigrate from Asia if possible. Australia has these wild vertebrate radiations like marsupial mammals, incredible things like elapid snakes and pythons, but also frogs and well, my favorite bird, kookaburra. Can never get enough kookaburra. You've got 250 species of frogs, 750 species of birds, over 1,000 species of squamate reptiles. So it's a really vertebrate-rich continent. But if we plot the species richness against the age of the crown of these groups for a whole bunch of different groups, what we see is a relatively strong pattern in richness increasing with clade age. It's not super tight, but it is there. But this is really a tale of two stories, in that these 4 clades are the only vertebrate clades terrestrial vertebrate clades that really have been with Australia since its separation, since it was part of Gondwana. And they display a really big diversity in the species richness. You have Keeled turtles, sorry, Arthur, they've not got that many species, but then the marsupial mammals are just kind of staggeringly rich. And then two other groups that fall in between. Pygopodoids geckos is things like diplodactylid, carphodactylid, and pygopod legless geckos, and myobatrachoid frogs. But when we look across all of the other vertebrate groups, terrestrial vertebrate groups in Australia, most of them are from Asia. So they're those orange points on this plot. Most of them have emigrated from Asia, and that's where the majority of the species richness, they've come in and then radiated once they're here, but they've come from a similar place. We also have a couple of groups that have the light blue things that have come from Africa. And at least one that we think that we're pretty confident has come from South America, which is the one I'm going to tell you about now. So across Australian frogs, they fall into four major groups. We have myobatrachoids, which are big, well, they're very diverse, and they're very old. I'll spend a little bit more time on them later in the talk. Hylid frogs or pilodryadid frogs, a single species of ranid that's found up north, and then microhylid frogs that we've seen Cape York and the top end. I'm going to focus on these guys for this first bit here. So we have across the frog tree of life, these four groups of frogs are really, of Australian frogs, are really dispersed across the tree. They're not clustered, they're not close relatives. They're really scattered across the tree, which makes for an interesting story in how they ended up here. So we used some next generation sequencing data, built a big tree, and started to look at the patterns we saw in the biogeography of the group. Biogeography and the divergence dating of the group. And what we see is that the three main groups, excluding the one that only has a single species, those three other groups have really staggered ages. The myobatrachoids are super old, 80 million years old for the crown. Their closest relative separated about 100 million years ago. The tree frogs are about 30 million years, medium old. And then the microhylids are two groups that have come from New Guinea in the last 10 million years or so. So they're really quite young. So this is really a stratified immigration or colonization of Australia. So these hylid tree frogs in Australia, we've got there's about 91 species in this family spread across Australia and New Guinea and surrounding islands. But they have a really amazing diversity. When you think of a tree frog, you think of big toe pads, big eyes, jumping around, maybe webbed toes, just like this thing up here, this Nyctomystes species. But Australian tree frogs have tried out everything else that you can imagine. You have mostly aquatic things like Litoria dallii in the middle, and down the bottom, Cyclorana, which are burrowing tree frogs. You don't see toe pads on those. They're not jumping tree to tree. Everybody loves Litoria dallii because these guys have one of the biggest testes to body mass ratios in vertebrates. So the males kind of float around on their testes like pontoons and call from the water. So these two are each other's closest relatives, or very close relatives, stuck in the middle of the tree here. So the morphological and ecological diversity has evolved very rapidly in this group into really discrete space and a morphological space. But the closest relatives of this Australian radiation of tree frogs are found in South and Central America and a bit in North America as well. But the closest family are these phylomedusid tree frogs. Some of them are also really impressive frogs that climb through the canopy, and you think of a red-eyed tree frog, they fall into that group. So we can take this tree and plot the distribution of these groups and start to fit biogeographic models across this entire tree. Except that some of the groups aren't that interesting. We already talked about the myobatrachoids, pretty confident they're Gondwanan. We actually even have fossils of their sister group, Calyptocephalellids, from Antarctica. So we know that they were probably spread across southern South America, Antarctica, and Australia. And on the other end, the microhylids, not that interesting either, except for the fact that they've come in from New Guinea more than once and roughly around the same time. But the tree frogs, we know the closest relatives are mostly in South America. But how did they end up in Australia? There's two prevailing ideas about how they got here. The one that we see in the literature quite a bit is this long distance overwater dispersal, which makes sense kind of timing wise when estimates of the group are a bit older, when the estimates are a bit younger. But when you think about the biology of frogs, that's a massive overwater dispersal event, and frogs are not very good with salt water. They're just little wet things, right? The alternative is that they've come from South America through Antarctica and then into Australia when these continents were either connected or very close to one another. And the timing roughly makes sense here. So we want to just keep in mind that over the course of the diversification of this group, these continents were previously, a single landmass and have been slowly drifting apart. But we can account for this in our biogeographic models. We can use these dispersal likelihoods, where dispersal likelihood decreases as a function of distance. So the farther away two areas are, the less likely you are to disperse across them. We can extend that by varying the likelihood across surface types. So dispersals across land, probably more likely than dispersal across open water, especially big stretches of open ocean. And when we do this, we can incorporate those into the biogeographic models, and we can test these two very discrete hypotheses. We can fix the stem lineage of the Australian tree frogs to either have been in Antarctica or to have been in South America before making this jump. So we can fix it in the two states. And what we find is relatively strong support for dispersal through Antarctica and into Australia, not a large overwater dispersal. These are obviously affected by these dispersal likelihoods, but you can try a number of values for these penalizations and you still get the same result. So this is what I like to think of as our Antarctic tree frogs. Maybe one day we'll if climate change continues in the way we're heading, we might find some tree frog fossils in Antarctica one day. The second story I'm going to talk about here is the idea of competition and competing with kin if necessary. I'm going to tell you about the best lizards, my favorite lizards, goannas. So in Australia, and where we have the greatest diversity of goannas anywhere in the world, monitor lizards vary in multiple orders of magnitude in size, not just body length, but also mass. They go from little pygmy things that would fit in the palm of your hand, all the way up to these Komodo dragons and parentes, things that would happily bite that hand off. And there was even a goanna that was even bigger than the Komodo dragon, a thing called Megalania varanus priscus, which would have cohabitated in Australia with Aboriginal peoples. This thing was maybe a meter or two bigger than Komodo dragons, which meant that this is a really monumental lizard. If you've been over to like the Canberra Reptile Park, they have a Megalania out front that is like the largest interpretation I've ever seen. I always say that it kind of grows by a meter every time somebody talks about it. And that one is like, I don't know, like 5 meters long or 6 meters long or something. But it was a big lizard regardless, and it would have been a really fearsome creature. So how do we end up with this amazing amount of variation within a single genus and within a single continent in Australia? Is this a result of character displacement through competition between species, forcing them into different niches? And if it is, aren't there other things that they might be competing with? There's other faunivores in Australia, particularly dasyurids and thylacinids that these monitor lizards would have been cohabitating with. And so there might have been competition between among goannas, but also with other mammalian faunivores. So I'm just going to give you a little context why we decided to chase this idea down. We took some linear measurements from across all carnivores in the world, or as many as we could get data for. And we included things like dasyurids, but also tigers, dogs, bears, things like that. And you can see in the blue dots are where the kind of goannas fall out on the first two PC axes. Dasyurids, yellow is the rest of the carnivorans, from everything from weasels to, well, we excluded things like seals because they've got a funny morphology. But one interesting note is that the Tassie tiger falls out squarely in the middle with a bunch of other dogs, which is actually, just more evidence that this thing was really so similar to canids. Anyway, all these groups, if we split this up by family, these groups tend to have different patterns in the major morphological axes of change. Bears are doing something particularly weird compared to the others. But dasyurids and varanids do pretty much the same thing, which is they keep shape relatively conserved and they increase in size. It's definitely true if you look at monitor lizards, especially for their attitude. You can meet a little goanna that's that small and it thinks it's a Komodo dragon. They'll thrash around and try and bite you and everything. And I've had the same thing with little planigales too. A planigale will try and bite your finger to get away. They're just remarkable animals. So they're really varying across size and not too much else. So when we look across the continent, if we were trying to find signature of this character displacement, we might look at individual communities of lizards. And if we're expecting that the diversity in Australian monitor lizards is not due to character displacement, that we might see populations or communities that look something like this, where they're structured maybe by close relative species, and they're similar sizes. There's not a variety of diversity. But what we actually see when we look across Australia is that the functional diversity of individual communities of monitor lizards is really spread. We have really small things cohabitating with very large things and medium things fitting in between. We also see diversity in their ecologies, but I'm not going to focus too much on that. If we take one step further, we see this functional diversity plays out on a scale between monitors and dasyurid carnivore, marsupial carnivores as well. So we built these models that I'm not going to spend much time explaining here, but we built these models where we had a phylogeny and a trait could evolve along it. We used biogeography to inform species that overlapped, even going back through time. And species that overlapped in range could influence the evolution of the trait body size across cohabiting lineage species. And that helped us generate these kind of matrices that we put into our trait evolutionary models. We can compare it to a whole host of other generic trait evolutionary models, like stabilizing trait evolution, stochastic Brownian motion style things, and other simplified competitive models that don't include biogeography. But what we find is the vast majority of support in model weights falls onto these competitive models. They're actually predict the evolution of the traits, the trait body size in goannas really well. So it fits the data very, very well. But that kind of falls apart a bit when we compare the monitor lizards and the dasyurids. Interclade is the idea that monitors and dasyurids are influencing the trait evolution of one another, of the two groups. We have relatively low support for that, the success of those models. But intraclade, there is high support. So there's strong support for the evolution of character displacement and body size in goannas and dasyurids, but separately, which kind of makes sense. We see dasyurids are generally crepuscular and nocturnal, and they're active predators, but the goannas are almost exclusively diurnal predators. So maybe they've partitioned this day-night thing quite well, and it's worked out for them all. Yeah, so these competitive models are heavily favored, and I'm gonna skip that. But I'm just gonna hammer home this idea of competition occurring in these groups in a slightly different way. And this doesn't have any analyses, though there's a paper behind this, but I'm gonna visualize this. So we've got, in New Guinea, we've got 23 species of monitor lizards, 20 of which are endemic. In Australia, we have 33 species of monitors, 30 of which are endemic. We share 4 species across these 2 regions, and there's 3 immigrants from New Guinean groups, clades, that have come into Australia, and there's 3 species that have evolved in Australia that have spread into New Guinea. But what we see is those species, when they cross this boundary, they're immediately running into a situation where there's already a number of species that overlap in their ecologies and body size and whatever traits they're competing in. And so species that have come from New Guinea and ended up in Australia fit into just these tiny marginal habitats around the fringes of the northern coast. They don't really make any headway into any other major habitat types. We have a kind of 1 or 2 mangrove-y type things, and then one thing that lives in proper rainforest canopy up in Iron Range. So we're seeing that this competition is playing out on multiple scales: an evolutionary scale within the continent that's resulting in the diversity of body size that we see, but also these species, once they get here, they don't speciate and they don't spread broadly across the continent. They're pretty much stuck in kind of marginal habitat around the fringes. Cool. I'm going quite fast, so we might be out of here early, but hopefully that's not a problem. The last story I'm going to tell you about is being flexible and to cheat if you've got it. I'm going to go back to that frog tree from the very beginning of this and talk about a different group. Instead of the tree frogs, we're going to talk about these myobatrachoids. And I've got a real fondness for them because they've done so many really wacky things, especially in reproductive biology. Up the top, you've got this hip pocket frog that once these tadpoles hatch, they fit into little grooves in the legs of the adults that carry them around. You've got the really scintillating turtle frog from WA that is just a marvel. And then 2 species of this gastric brooding frog, Rheobatrachus, which have unfortunately both got extinct. But the females would swallow the fertilized eggs, and then the young would develop in their stomachs. So myobatrachoid frogs have tried everything you can imagine in reproductive biology. And it's worked for them so far. I'm going to focus on this small group here, which includes Neobatrachus and Helioporus. So they're 2 genera that are sister one another, and they share a common ancestor somewhere around 45 million years ago or so. But it's really a tale of 2 very different stories in where they live in Australia. Helioporus has this kind of Gondwanan forest distribution that we see from really mesic things that haven't adapted well to the change in climate that's happened across the continent over the past kind of 15 million years or so. And Neobatrachus are kind of the inverse of that. They're one of the rare frog groups that have done really well in arid biomes across Australia. And this is likely to do with this amazing physiological development they've had, which is they make epidermal cocoons. Both of these genera of frogs burrow into ground, sand or loose soil, but Helioporus or Neobatrachus create this cocoon that keeps them moist and protected from really dry environments outside. And they can sit in that cocoon for 6 months, 12 months, 18 months, while they're waiting for big storms to come back, so moisture to come back into their environment. And this has really allowed them to take advantage of places where rainfall is really unpredictable in a way that Helioporis cannot, because they can't create these cocoons. And this is just to show you what I showed you, the spread across the continent, but this is what the species richness looks like, where you have a kind of increase in richness in Western Australia. So as part of a different project, we looked at the phylogeny of this group. We have 9 species. They're pretty cool looking frogs. I like them. You know, they're a sizable size. They have a nice call. They have nice calls, I guess. But there's something more interesting happening. People that looked at the karyotypes of these a while back in the 80s, they identified that 3 of these 9 species are actually tetraploid. And when we build our phylogeny, we identify that those tetraploids aren't a clade. They're not closest relatives. They're actually 3 separate whole genome duplication events through we assume, or we believe, that it's autopolyploidy that's a spontaneous event instead of allotetraploids. So we start to try and reconstruct this species tree, and we look at all of the individual gene trees. We have several hundred gene trees to look at. If you use multidimensional scaling to look at the distances in multidimensional space between individual gene trees, we find that there's actually at least 2 clusters of gene trees that are giving us 2 different species trees, one that looks like that and one that looks like this. So we looked at that and we said, that's weird, and it's maybe some sort of hybridization event. Or there's introgression between species. We already know that there's introgression between some species because we've identified triploids. People have collected triploid Neobatrachus frogs before, so we know something weird is happening. When we look at when we use any of our population genetic assignment software, we identify that all of the diploid species are pretty kind of pure clusters. But the tetraploids are really messy mix-ups of gene flow that have come from a number of different places. That's just a dated tree for the diploid species. So we know they're kind of maybe end-Miocene-Pliocene group. And when we dig down deeper into the introgression, the patterns of introgression, what we're seeing is a lot of introgression between the tetraploids, but also flowing from the diploids into the tetraploids. And we have strong evidence for at least 1, 2, maybe 3 individual introgression events, but this is obviously a very stochastic, tough thing to estimate, especially in a system that appears so messy. Okay, so we have 3 tetraploid species, we have 6 diploid species, and we're looking at how they're spread across the continent now. We know that they can extend into these arid areas, and we kind of wondered how they've been able to do this over the climatic change. And so we estimated their distributions across the continent, or estimated suitable habitat since the last glacial maximum to present. And one thing you might notice is that ranges for some of the diploids have really shrunk down since the last glacial maximum, whereas the tetraploids have either increased their suitable habitat or remained about the same. When we look at the genetic diversity of the diploids, and we use Tajima's D as an estimate of this, a very rough estimate of this genetic diversity, we see this relationship between change in suitable area. So decreases in change in suitable area are also linked with decreases in genetic diversity. And you can see in this Neobatrachus pictus over here, this is a really kind of strong signal of climate suitable habitat change. But when we throw the tetraploids in there, at least 2 of them show very little contraction in suitable habitat, and they show a signal of expanding population or genetic diversity. And we suggest that this is probably due to them picking up diversity through these introgression events between each other or with the diploid species. So in this situation, these introgression events are potentially buffering the tetraploid species from this effects of climatic change that's been happening on this kind of glacial scale. That's actually, I think, all I'm going to talk about today. I didn't want to stretch too long. So I've just given you a few choice stories about how you can succeed in Australia via immigration, being an immigrant myself, competing, and then being really flexible. I just want to kind of end with not a plea or anything, but just a suggestion to go out and appreciate the amazing diversity of animals and plants in Australia. I don't think I'd ever seen a wild parrot before I moved to Australia. And Canberra, they're literally like in my gutters and raiding my garbage bin. It's like, it's amazing. So we've got it really, really good here. So appreciate that amazing diversity that, you know, lots of people will never, ever get to experience. These projects are the result of some really fruitful and fun collaborations I've had over my PhD and postdoc. So I want to, first postdoc, so I just want to thank a bunch of these people that I have worked with. Particularly Scott and Steve have been great people at facilitating these projects. And I'm currently funded by European Commission on a Marie Curie Fellowship, but have had a lot of support from ANU and Bioplatforms Australia in developing workflows to facilitate this research. If you don't believe any of these tall tales I've told over the past half hour or so, these are the papers that those stories came from. Feel free to look them up if they're of interest, and you can get a bit more nuance, learn a bit more about the methods if you're interested. But they were all projects that were really collaborative and a lot of fun. So I think I'm going to call it there, and hopefully you guys enjoyed that very light gloss over a few ideas on how to succeed in Australia.
Thank you, Ian. That was great. Do we have any questions?
Thanks. I'm sure that was good, thanks. I'm sure you covered off in your papers, but I was curious, a lot of the varanids, some of them have really big ranges.
Yeah.
And so there's a potential for at one end of their range to be competing with one group of species in a different subset at the other, which obviously then has the potential for inconsistent outcomes in terms of how they might be evolving.
Yeah.
And so I'm just curious how you dealt with that, or
we didn't have a specific way of dealing with that, but it's funny how their ranges generally track their body sizes. So you have lots of the small, really small kind of dwarf goannas are kind of short range endemic-y things in the Pilbara and Northern Territories and Northern Queensland. And the larger goannas have these much larger ranges, and there's fewer of those real large species. I can't say we had a particularly clever way of dealing with it, the exact solution or the exact problem that you're mentioning. But we hoped that by incorporating these ranges, it was generating a more kind of holistic view of the evolution of these traits. So there's definitely room for expansion of these things. The one big shortcoming, I would say, to be overly honest, is that these trait evolutionary methods that we use very rarely incorporate intraspecific variation. And that could either be a real benefit or a real downside. Sorry, not excluding it is never going to be a benefit, but including that information can either be beneficial or not. Sometimes we see a species that has a really broad range also has great variation in the trait of interest, depending upon who it's overlapping with at those extents of the range. And they can be really good indicators of character displacement happening. And we do see that in some of these things, but that's not captured by the models, unfortunately. So, thanks for the question.
Thank you. Great talk. I have actually two questions. The first one was about the integration into those two species of frogs, because those are tetraploid. And did anyone saw any sort of like check whether there was a sex chromosome turnover event in those? Because integration actually plays a vital role over there, and in sex chromosome turnover, whether it happened or not. So that was my first question. First question, and the second question was the route you mentioned in the first one, that the frog coming from Antarctica, not from that way. So it sort of like they stayed in the Antarctica for a while, right? Because like there has to be a genetic signature in those. Was it evident?
All right, I'll try and answer them separately. The first question was about the sex chromosome turnover. It's a super cool group that I really enjoyed working on, but I'm not a genomicist. So after that project, I've left that up to Paulina, who is my co-author on that. And she's doing some really, really cool work on the genome evolution of that group. So they have really great data. And I think you'll probably hear quite a bit more about that in the next few years. She's trying to kind of develop them as a model system for understanding polyploidization and the effects on genomic architecture. So I think you'll hear more about that. I don't have an answer about the sex chromosome turnover. The second question about the signature in the hyalid frogs, we have a clade of frogs, the phylomedusids in South America, and we have a clade in Australia and New Guinea, but we have nothing in between. So everything in between is always going to be a bit hand-wavy. This was our attempt at trying to answer that in a structured and visible way. And we're presenting a hypothesis. We're just presenting a hypothesis that we believe we have some more support from the models. We'll never know if they, fell out of a palm tree and ended up in Australia, or if they did come through Antarctica, unless we, we're lucky enough to find some fossil hyalids in Antarctica. But it's tough work down there. They do turn things up every once in a while, but just don't know. Did that answer your questions? Yeah. Cool.
Yeah, thanks. That was really interesting. With the tree frogs or hyalids, how certain are you of the date of 40 million years? Is that presume that's 40 million years plus or minus some number, and then presumably the geologist would be in terms of when exactly the last land bridge left would be plus or minus some number. So, and what I'm thinking, if that's like maybe a little bit older than 40 million years, and their best estimate of when the last land bridge left is maybe not quite so accurate and all that sort of stuff, that maybe they could just literally hop through those continents.
Yeah.
But also the other thought occurred to me is maybe they started in Antarctica and then one way went into Australia and the other way went to South America, and there's probably not a good way of, did you model that possibility?
Yeah, so there's kind of two parts to that question I'm going to try and unpack. The first is related to the confidence and divergence estimates. And I trust the divergence dates about as far as I can throw them, which is not very far. The divergence estimation or diversification estimation is like always going to be fraught with problems, especially you get a huge variation in estimates for the age of the group. Our estimates are the best we can do and incorporating more fossil information than has been done previously. And it puts the clade slightly older, about 5 to 10 million years older than previous estimates or most recent previous estimates. But those 5 or 10 million years can make a big difference in your models for your biogeographic inferences. So we tried to use a series of trees that were of different ages and refit models to see, what's the breaking point? When does it flip over from dispersal through Antarctica to a dispersal across water. And that doesn't happen until the most common ancestor between the Australian and South American clades is much younger, down 30 million years ago or so. I just distracted myself in responding to that. I can't remember, but I guess the geology matches up roughly with the timing. Antarctica and South America were linked, if I remember my geology correctly, were linked slightly longer. Australia separated off somewhere around 40, 45 million years ago. So they still would have had to make a small kind of over water dispersal across from Antarctica into Australia, but we include these things in our models. Yep. Yeah. And that, to me, that seems more parsimonious. But,
Or did you
sorry, there was a
consider that
second to
they
the question.
burrowed through the earth?
Yeah, they could have gone straight from Patagonia into Perth.
Pipe up online as well if you have any questions, or just put it in the chat.
Oh yeah. We didn't model that as a process, but I kind of, if I can, it's okay. Don't worry. No, don't worry about it. It's totally fine. The tree shape is such that the pelodriatids are kind of sister to the South American group, which is sister to a group from South, Central, and North America. And so in the models, you'll reconstruct the origin of the hylids as being in the Americas. So we didn't anticipate that an Antarctic origin was probable. So we didn't try that out, but maybe it is worth testing. I don't know. Like I mentioned before, though, this is another one where we're never going to have a good answer for that. But it's possible, I guess. Gondwana was a big mix-up, and the habitat was suitable for things. 30 million years ago or so, we know that it was, or 35 million years ago, it was quite wet and temperate. So it would have suited them.
I think I have a question. I'm not 100% sure, so hopefully it emerges. So when you were doing your modeling of trait evolution, you had ranges included in that as well. Could you look at, so as different traits evolve, can you link that to the SDM and, find areas where they should be able to persist, but maybe there are particular traits that are kind of pushing different groups out, and are they like consistent?
In the goanna project?
Yeah.
The distribution modeling is a tough one because that's like a bit outside my area. So what we did is we used a very generic method for estimating the ancestral ranges. We just used kind of a Brownian diffusion model that takes the contemporary ranges of species and then kind of tries to work its way back. But if you had proper distribution models, you could do it at a finer scale for sure. And I think, but also we don't know what the anagenetic change in climate preference might have been either. I don't have an answer for your question, Robin, but I think it's a cool idea. There have been people that have tried to work on this idea of the ranges and climate preference influencing traits in a single comparative framework. Michael Landis and Ignacio Quintero have worked on this quite a bit, and they have some really cool papers on testing these ideas. But they're really intractable for large groups. They work well in island systems where groups are kind of discretized. So people have tested this on Hawaiian things and anoles in the Caribbean, but on a continental scale, it becomes a bit more diffuse and harder to nail down.
Well, if there aren't any more questions, join me in thanking Ian again. we're going to head out for lunch. So if anyone wants to join, you're welcome to.
Thanks for coming. I appreciate it. Thanks for that.